Hello, everyone. I'd like to welcome you to Life Science Across the Globe. And today we're very pleased to have our sister institute, Cold Spring Harbor Laboratory, for their second round of talks. And I would like to introduce the president and CEO of Cold Spring Harbor, Bruce Stillman. Bruce's professional and scientific career is really marked by at least three terms in my opinion: dedication, focus, and accomplishment. So first of all, with regard to his professional life, Bruce started his career at Cold Spring Harbor Laboratory as an independent investigator in 1979, and he's been at Cold Spring Harbor ever since and has led Cold Spring Harbor as its president since 2003. And during that time, not only has he continuously grown Cold Spring Harbor's reputation in the science that it produces, but as you'll also see in the cultural talk, also has made Cold Spring Harbor a key intellectual center and a meeting ground and effectively a thought center for many aspects of the biological sciences, and has also led efforts in science education. And for those of you who did not see Cold Spring Harbor's first talk, I encourage you to see the video recording on their DNA Learning Center, which was a remarkable effort in science education. Bruce's also scientific work has been characterized by dedication, focus, and accomplishment as well. He spent his scientific career trying to understand the mechanism of DNA replication, and really in a remarkable research program kind of systematically dissected the mechanism using biochemical and structural approaches, and for which he's received many awards. And it was actually very nice to be in the same class of the Canada Gardner Award a couple of years ago, where I got to know Bruce better and his work. So with that, I'd like to give the podium now to Bruce, who will introduce today's speaker.
Thanks, Ron. It was actually a great pleasure to be able to travel around Canada with you last year. I want to also thank the Janelia team for putting on this series. It's been very successful, and I want to welcome you back to Cold Spring Harbor Laboratory. Today, we have two speakers, one science and one in the culture series. In the science area is going to be Zach Lippman, who's actually happens to be a graduate of the Cold Spring Harbor School of Biological Sciences, where he did his PhD with Rob Martinson. And then he did postdoctoral work in Israel with Danny Samir. And we were very fortunate to be able to recruit him back to Cold Spring Harbor in 2013, where he's now a professor and Howard Hughes Medical Institute investigator. And as you'll see, he's working on some really, truly innovative plant genetics, and that I think going to have a global impact on agriculture. And in recognition of this year, he won the very prestigious National Academy of Sciences Prize in Food and Agriculture, which is quite an achievement. So I'm going to turn it over to Zach, who will give the science presentation.
Great. All right. Thank you, Bruce. And also thank you to the organizers. Happy to be able to share what we've been doing in my group at Cold Spring Harbor for the last 11 years or so. And it does relate to this title quite specifically, as you'll see, that we're very interested in leveraging what we're learning in fundamental plant biology to agriculture as it relates to genetic control of productivity traits. The image that you're looking at here is actually a field from this summer, not too long ago, a field not too far from where Rebecca spends most of her time, near Banbury. And this is our tomato field. And every year we have about 4 or 5 acres where we're using this space to essentially gather quantitative data on various productivity traits. And so you'll see quite a bit of data coming from these past fields and also this current year's field today. Okay. So to give a background of what we do in the lab, it's really based on these 4 topics. So we focus on development, and from the developmental perspective, we're looking at stem cell control in plants, particularly as it relates to these structures called meristems, which is where stem cells reside, and stem cells mature from vegetative to reproductive states over the course of life. And we're interested in that reproductive transition and the stem cell signals that moderate that. That leads into questions of how stem cells and their maturation affect evolutionary diversity as it relates to the process of flowering and flower production on these structures called inflorescences. We're also interested in looking at how this relates to domestication of plants, not only in tomato, but in other crops as well, but primarily tomato. And here's just an example that you'll hear about a little bit later on how domestication was important and also the improvement of tomato as it relates to modification of plant architecture. And then what we're learning from development, evolution, domestication historically has really been informative in how we develop new principles and tools for engineering improvements in crops. So just to give you a backdrop of the larger questions that we're interested in has to do with how genetic variation is, of course, the driver of crop improvement. And historically, I think many would be familiar that this is coming from two sources of genetic variation. There's natural variation, which we would get from nature's mutagenesis, where you have domesticated plants that have variation that exists already endemic, as well as from wild ancestors. Now, this variation is quite widespread, but it's random and it's quite slow and cumbersome to use in terms of a breeder's perspective. About in the 40s or 50s, there was an attempt to use induced genetic variation from chemical mutagenesis or irradiation in order to enhance the variation that existed both genetically and trait-wise. Now, this worked to some extent, but it's also quite random and it's also very slow and inefficient. And we're now in the age of where we have the ability to use genome editing to make targeted changes, which is much more fast and efficient compared to natural and induced. And I want to point out that what's critical here is that what we're learning from genes that were important from natural modifications and traits during evolution, domestication, and breeding, as well as induced mutations, these genes that we're discovering are informing us of the targets that we now want to use for genome editing. The lab is really interested in flowering, and taking a step back from some of the details I'll tell you in a few slides, one thing I think is important to emphasize here is that flowering and the transition to flowering has been responsible for multiple agricultural revolutions. And it has to do with a toned down version of a system of flowering that involves two protein hormones known as florigen and antiflorigen that work in opposition or in this yin-yang relationship to promote from florigen and repress flowering from antiflorigen, which gives you a naturally ecologically selected advantage in terms of flower production. And then, of course, modification of this would then be very critical for agriculture because it would change not only the number of flowers that are produced and then fruits and seeds, but also the architecture of the plants, as you'll see in several slides. And if we look at just these examples of traits here, each one of them, each one of these plants, I should say, has had modifications of multiple traits as it relates to modification of the yin-yang balance between florigen and antiflorigen. And just to give you two examples before getting into our work on tomato, there have been major changes in soybean and cotton, where you can see on the left images for both crops, these very large, tall, and in case of cotton, bushy plants, that when you have a modification of the florigen-antiflorigen system, where you have now a bias towards florigen or flower promoting signals, you end up with a faster flowering transition, which matures the plant faster, which ends up resulting in a more compact plant, either from the whole plant perspective or in the case of cotton on the side shoots, which gives you these modifications that are beneficial for agronomic purposes, for example, for higher density planting, which is essentially what these soybean and cotton determinate plants allowed, coming from the ancestors of indeterminacy, which is continuously growing. There's a similar situation in tomato, which is our system of choice, and it has to do with modification of primarily plant architecture, where tomatoes, if you've grown them in your garden, you've noticed perhaps that many of the varieties have this continuous, very tall, lanky, bushy growth pattern, which is typical of the wild ancestors of tomato as well as the early domesticated forms. But what happens is when you have a mutation, which is a classical mutation, in a mutation in the promoting the flower repressing hormone antiflorigen, you get an effect that is, as the mutant is called, causes a self-pruning impact on shoot architecture. So if you look at the red arrows, you can see that instead of having a nice spaced out continuous growth of these clusters of flowers known as inflorescences, when you have a self-pruning mutation, you get this burst of flowering that occurs on a much more compact plant. And this is because by removing antifloragen through a natural mutation in self-pruning, you're shifting the balance of this relationship to flower promoting signals, again, causing a faster maturation of the stem cells, leading to this overall plant architectural change. So this resulted in the reason why we have this row production of tomato that you see here from an image in Florida, giving you a compact architecture, again, the burst of flowering and fruit production, and then of course, becoming a row crop like you have with corn and soybean and cotton. Now, I want to tell you that this is not such a simple story. This mutation was discovered in 1927, and it was immediately recognized for its agronomic potential because of its compact architecture, which would facilitate harvesting and also high density growth. But if you look at these videos on the left, which I hope are coming through reasonably well, there are two major types of tomato production. There's processing tomato production, which is harvested with a combine, as you see. And then there's fresh market production, which is grown towards quite large plants, considering what you see in the processing tomato production, and it's harvested by hand. Now, processing is primarily occurring in California. Fresh market is primarily in Florida. And what the key point here is that both of these varieties that are grown, in fact, all the varieties that are grown for these two types of tomatoes, carry the self-pruning mutation, but they look quite different in terms of their overall determinate architecture. And this is because over time, there was a requirement of very time-consuming, laborious tuning to modify quantitatively the architectures and the yields to suit the particular agronomic needs. Now, I don't have time to show you all the data, but a number of years ago, we showed that we could predictably tune the modification of architecture and yield in a much more efficient way compared to how breeders have done this in this very slow, laborious way with selection, using a series of mutations that quantitatively reduce the dosage of florigen, such that as you reduce florigen, you're taking away some flower promoting signals in this self-pruning background. And that effectively allows the plants to slow their maturation and get slightly larger in a quantitative way, which allows more clusters of flowers and fruits to form on still compact determinate plants. And so this is to show you that a series of mutations of various allelic strengths in florigen and an interacting protein partner can give you a quantitative, predictable tuning of the type of architecture that we already see in growth conditions in Florida and California. So this really opened the door to what's possible for quantitative tuning using a combination of both natural and induced mutations. And I should say that these mutations did not come out of primarily CRISPR mutagenesis at the time. It came out of the mutagenesis that we use typically over the last 20 years, which is chemical mutagenesis, EMS to be specific. And so we had to search for a lot of these mutations in large fields using a classical mutagenesis screen, which is really not that efficient. With CRISPR, we've been able to take this to a new level. This is now looking at a very extreme opposite end of the spectrum change in plant architecture for tomato to be grown in these very confined conditions, which is urban agriculture. And so this is a company in Boston, Massachusetts, where we collaborated with them to grow our modified tomatoes that now, instead of becoming a slightly less compact and higher yielding, we can make them ultra compact and plant them at extremely high densities where the yield per plant is reduced, but the high density allows us to grow these plants in essentially shipping containers and these vertical farms that you see here. So we can capture the whole spectrum of quantitative variation using both natural and induced chemical and genome edited changes in these flowering regulators. So that's plant architecture. And I'd like to tell you a little bit about another agricultural trait that's important for productivity, and that's the inflorescences themselves where the flowers are born. And this is just to show you that in tomato, being a member of the larger Solanaceae family, you have remarkable variation in the architectures of these flowering shoots known as inflorescences. And compared to tomato, which I'm sure many of you have seen, grows on a single branch with multiple flowers along a linear branch. you have a whole series of quantitative and qualitative variation in the Solanaceae family, of which tomato is a member, from very extreme branching, like you see in this species on the lower right, to very, very weak inflorescence branching, like you see in this tobacco species, which produces just one flower. Tomato will make 6 to 7 flowers on each inflorescence, and this will occur recurring throughout growth. And then there are wild tomato relatives, which will branch much more subtly. For example, Solanum pruvianum, which comes from South America, will make 2 or 3 branches on each inflorescence before it actually stops producing flowers. And this is really, I think, an indication of the type of quantitative variation that could potentially be achieved in a system like tomato, where we know that we can get mutants that have very extreme branching that mimic wild Solanaceae species. But the problem with such extreme branching is that on the surface, while it looked like having this overproliferation of these stem cells and meristems, as you're seeing here in these middle lower images due to a slower maturation process, which translates to many more branches and flowers. While this may look desirable on the surface because of all those flowers that are produced, you get this imbalance in growth. And you actually have lower productivities because the plant is so focused on making these branches and flowers that the energy resources are imbalanced and you don't end up setting any of these flowers efficiently into fruits. Now, these are a series of mutations that we've worked on over the years and identified the responsible genes. This one in the middle is a homeobox mutation. This one on the bottom is a double mutant in 2 transcription factor genes known as MADSbox genes. Now, what we would really like to be able to do is take away that extreme branching and get something much more subtle like Solanum pruvianum, where you have a slightly relaxed and slower maturation, which gives you a more balanced production of branches and flowers. And compared to the wild ancestor in most varieties of tomato where you don't have this branching, it would be something that would be desirable from the perspective of productivity because we don't have any variants that are easy to work with to give us the type of branching that occurs in the wild. Now, you might say, well, why don't I just do genetics with pruvianum and some of these modern varieties or even the wild ancestor? There are genetic incompatibilities that prevent that from happening. And so what I can show you on this slide, which summarizes many years of data, is that knowing, again, some of these mutations that give us extreme branching, which is undesirable, we can start to make modifications using both natural and genome edited mutations in these transcription factor genes to get, similar to plant architecture, a modification of dosage of these transcription factors, such that as you go down in the dosage among redundancy among 3 of these genes, you progressively increase the severity of branching such that you get an extremely undesirable case of what looks like cauliflower when you have this triple mutant. But what I want to point out is that by playing with these mutations, both in homozygous and heterozygous conditions, we can tune the dosage levels in such a way that we can find these sweet spots where branching is exactly where we want it in terms of its subtlety, where you don't have this imbalance of vegetative and reproductive growth. And I don't have the time to show you the data, but collaborating with my former postdoc advisor, Dani Zamir in Israel, we're able to use this particular genetic constitution to increase yields in greenhouse tomatoes that are grown very tall plants and very compact in terms of their density, and where you only have the ability to improve yield based on increasing the branching in the inflorescences. So this is nice to be able to see that we can tune these traits quantitatively by playing with dosage, and in many ways playing with epistatic relationships among florigen and antiflorigen, as well as the redundancy among these MADSbox transcription factor genes. But over the last few years, we've been wondering if there are ways where we can tune traits much more efficiently. And what was apparent is that while we had the ability to generate coding sequence mutations by CRISPR, and to take advantage of natural mutations that were fortuitous in what arose through just screening through natural variants or through EMS or chemical mutagenesis, that we were limited in the type of alleles that we had. And in particular, we needed many more alleles that had a range of phenotypic effects. So as a geneticist, really what you're boiling this down to is saying, I want an allelic series. Now, an allelic series can do quite a bit for you when you think about it from studying fundamental developmental questions. But from the point of agriculture, it's quite clear that allelic series already exist, and they're very, very valuable for quantitatively tuning traits where nature has already sort of given us certain levels of dosage changes in genes that give us the type of trait modifications that would be useful for particular crops that you might be working with. So here I'm going to show you an example of tomato from a number of years ago where we collaborated with a group at the University of Georgia to show that a natural mutation in a cis-regulatory region, in this case, the promoter of a stem cell regulating gene known as clavata3, gives you a moderate allele where you don't completely disrupt the entire function of the gene, such that it results in an increase in the number of sections of seeds in the fruits known as locule, but not in an extreme way. And this results in increased fruit weight. One of the major drivers of increased fruit weight that occurred from tomato being these small berries in the wild to the very large varieties that we have now is in fact an increase in these locules that you see here. And this particular mutation gives you probably an increase of from 2 to 3 locules all the way from 7 to 10 locules, which of course would increase fruit weight proportionally. Now, the reason I'm showing this particular allele and relating it to this desire to have an allelic series with a range of quantitative effects due to changes in dosage is that we recognized quite early on that cis-regulatory changes, as opposed to coding sequences, would be a more practical and efficient way to achieve the type of dosage changes that we want because it's modifying expression levels. Especially with genome editing and especially in plants, making particular coding sequence amino acid changes and knowing which ones we would want to make, first of all, is limited based on the tools, as well as the mutational space being extraordinarily high. Which amino acids do you change to get which particular quantitative modifications is not clear at all for any gene. So in this case, we said, well, let's not mutagenize the coding sequence. Let's do a multiplex bashing or mutagenesis in vivo of promoters of genes. And in this case, the clavata3 gene, where we would hope to mimic the natural inversion effect, but also to create an entire range of alleles that have a range of phenotypic effects, with the hope that this allelic diversity would translate to the type of phenotypes that you would use in breeding. And so I'm showing you here, over many years, nearly 30 alleles that were created by a postdoc and a student in lab, Xingang and Lindsay, where we have a whole collection of large deletions shown here at the bottom, even inversions, all the way up to very weaker effects where you have small indels. These are quite difficult to look at in this particular context. So what we've been doing now is encoding them in 20 base pair bins where you can see that the deeper purple reflects a greater perturbation, in this case, deletion in purple or inversion in red of each 20 base pair bin. And the reason I'm showing you these alleles is that, in fact, this worked beautifully in the sense of being able to not only mimic the domestication natural inversion of this gene clavata3, but also to create a complete continuum of quantitative variation for locule number. The other thing that I think becomes apparent is that you can start to see how you can learn how particular regions of the promoter and perturbations of those regions can give you rough estimates of predictable outputs. In this case, if you're perturbing the proximal region, you very subtly engineer QTL, quantitative variation, where you bump locule number from 2 to 3 to about 3 to 4. If you go distally, you start to bump locule number to about 6 to 8. If you start to perturb both proximal and distal regions of the promoter, now you get your more severe phenotype that matches the coding sequence knockout alleles that we've created. And so the question was now, well, is there a way to design some rules that would explain phenotypic outcomes? And can we now look at dissection of the cis-regulatory control and complexity, not only from a fundamental point of view, but also from the ability for us to be able to make targeted mutations in promoter regions to get predictable quantitative tuning? So for this, we have another gene that we turn to, this homeobox gene, to get a deeper characterization of cis-regulatory regions and see if we could get predictable tuning. And I just want to step back and say that the way we've been looking at this in collaborations with a group at UMass Amherst and also Hebrew University is to ask whether we can look at some of the chromatin features and also sequence features that might tell you particular functional regions of the promoter that might be important compared to others. And what this amounted to was taking several genomes in the Solanaceae family and looking for conserved non-coding sequences in promoters. We have open chromatin data from ATAC-seq, And to summarize quite a bit of data, I'll just tell you that we have quite a bit of overlap, as you might expect, between conserved non-coding sequences and open chromatin. And what I think is really nice is this graph, which was done by Anat Hendelman, a postdoc in the lab, showing that if you look at the attack-seq windows for these open chromatin regions, and you look at the midpoint of that particular peak that was called, the density of conservation is highest at that midpoint. So it really tells you that conserved non-coding sequences are a proxy for open chromatin and likely functional regions of the promoter. And so this is looking at an extended version across 8 Solanaceae species of sequence identity and similarity in the promoter region of this homeobox gene known as WOX9. And zooming in a little bit closer, you can really see that these blocks reflect many ways a functional extension of the gene. So we're looking beyond the coding sequence, which of course you have identity, but even some of these regions quite distal, about 4 kb upstream, show that you have even higher identity than some of these coding sequence, regions of coding sequence that you have across these 3 species. And this represents, relative to tomato, about 30 to 40 million years of evolution. So I'm going to summarize quite a bit of data in one slide and say that we've done a similar thing as we've done for locule number and the CLABATA3 gene in tomato. And now we've mutated the promoter of this WOX9 gene, this homeobox gene, and created about 25 alleles, several of which are shown here. And what I want to point out is that what you can see is that as this distal region is perturbed, now is where you start to get this nice weak branching, which mimics the type of branching I showed you earlier, where we were playing with the natural and induced mutations in those transcription factor MADSBOX genes. Now, what's really nice is that it was quite clear that this region overlaps with a conserved non-coding sequence and open chromatin. And so it points to this particular section of the promoter to target in order to get this moderate branching. Now, this particular large inversion, which extends well upstream, has more severe undesirable branching. So now we're not only able to see the particular region of the promoter we'd like to target for mutagenesis, but also the particular subregion that might be critical for us to get the right sweet spot in dosage to get the quantitative branching that mimics the wild tomato species from South America. So what does this all mean in terms of a broader perspective? I've told you quite a bit of data about tomato, and I told you that this is relevant in the context of breeding of tomato and quantitative tuning using genome editing to modify dosage. But what does it mean for all these other traits? Well, I think it relates to natural variation again. So if we look back at some of the studies that others have published, as well as our own work over the last few years, if you're using long read sequencing now, you're revealing, of course, not only single nucleotide polymorphisms, but also these large structural variants that are changing naturally in the genome. So this, of course, includes deletions and inversions and larger rearrangements. And so working with Mike Schatz at Johns Hopkins University, we published this past year sequencing by Prometheon, long-read nanopore sequencing, 100 genomes, which revealed 230,000 structural variants across these 100 different varieties. Now I'm going to take a moment and tell you that this is really, in my mind, reflecting what I would call cryptic genetic variation. I think others might not completely agree with the classical definition the way I see it. Cryptic genetic variation is typically termed as variation that only reveals its phenotypic consequences in the context of other mutations. But I think it also applies to very subtle changes that would be extremely difficult to map and identify the responsible gene. In other words, very, very weak alleles that are very difficult to detect in the context of all the other segregating mutations that exist in a population. And so these cryptic alleles are clearly going to be important in natural variation. We know that they are. We found many, many thousands of them. And we know, looking at particular structural variants in these 100 genomes, that 40% of them reside within the nearby genes. And among those 40%, 80% are within proximal regions of genes, the 5' and 3' cis-regulatory regions. And of those, 10 to 20% are associated with expression changes. And so this is this huge reservoir of natural structural variants. Deletions make up a large majority of these, as do inversions. And so it relates to this question of, well, what about these implications for these structural variants and quantitative tuning of dosage that have occurred naturally, not only in tomato, but across all these different crops that have been subjected to selection through domestication and breeding? And to what extent can we now leverage what we're learning from engineering structural variants in regulatory regions and tie that back to what we can then learn from natural structural variants that have been provided over thousands of years of evolution? And so hopefully in the question and answer, I can address some of these larger questions that relate to this relationship. But with that, I'm just going to say thank you to the people that have done the work, particularly Xingang, Lindsay, Sophie, and Anat, postdocs and students in the labs, as well as many other people who have contributed in various aspects of the projects. Don, Madeline, Noah, and Julin Wu have been important in the cis-regulation work that I talked about as it relates to CRISPR, and we work extensively with Joyce Van Eck at the Boyce-Thompson Institute on our genome editing in tomato. And thank you, of course, to funding agencies, including HHMI. Thanks very much.
Thank you, Zach. A very elegant study and a fantastic talk. So clearly, I'm not the only one enjoying the talk because we have some interesting questions from the audience. So the first question comes from Cao Dong. Cao, please unmute yourself and ask your question.
Okay, thank you for your talk. Can you hear me?
Yes.
I do.
Yeah. I have a question about the CRISPR tomatoes. Are they considered as genetically modified crops, genetically modified organisms? And if yes, how are they regulated?
Yeah, very briefly, it depends on where you are in the world. So in the US, they are not considered classically genetically modified. So the regulation is much less reduced. You simply have to report it and then they give approval. But for example, in other parts of the world, for example, in New Zealand, they're still looking at this as genetically engineered and the regulations are very strict. And the same is true in Europe. And so right now it's a hodgepodge of not being regulated and being regulated. And the hope is that over time, it'll be disentangled from the classical genetic engineered crops and it will not be under as much regulation.
Okay, thank you. I have another small question. Is how do you control the off-target effect of the CRISPR?
Yeah, in plants, it turns out that off-targets seem to be minimal to non-existent compared to mammalian systems. And there's been extensive data over many, many model and crop plants to show this. You can periodically find off-targets appearing, but for the most part, you really don't find off-targets. And the nice thing about plants and crop breeding in general, leveraging genome-edited mutations, is that if you do have off-targets, the natural process of breeding and what's called introgression basically eliminates them through Mendelian segregation. So unless those off-targets are tightly linked to your target gene that you've modified, you can remove those within one generation.
Okay, thank you.
Thank you, Zach. The next question is from Thomas. Thomas, please go ahead. Unmute yourself and ask your question. All right, in the interest of time, I'll read out Thomas's question. Thomas wants to know, do these CRISPR mutations in the coding regions have effects only on the level of RNA expression or also on isoform types in different and or prevalence of overlapping non-coding transcripts?
You know, there isn't a whole lot of data out there, and it's probably because there aren't a whole lot of genome edited coding sequence or regulatory mutations that have been published in plants. We've done quite a bit of RNA sequencing on our coding sequence and regulatory mutations. We have not dived deep into finding whether isoform changes are occurring or if they affect specific isoforms. I presume it's probably happening in rare cases, but for the most part, coding sequence mutations cause frame shifts and you end up with a knockout mutation, and regulatory mutations are affecting expression levels or where and when genes are expressed. And so isoform changes, you'd really need to start looking at particular splicing variants to see whether or not that's happening. And that's something that hasn't been looked into deep yet.
All right, thank you. So the next question is from Jason Lin. And again, I think I'll read out this question. Jason wants to know how much of the structural variation between those tomato genotypes is related to transposons or repeats.
80%. and it's probably an underestimate in the sense that we know that if we annotate those structural variants, the sequences themselves annotate 80% of the time to transposons and other repeats. But I would argue, and I think this is in the literature to support it, and we can see this if we look at sequence context, that even those structural variants that are not annotated as transposons, they are often in transposon environments, which are facilitating the type of structural variants that you see. And so this could be because of rearrangement that occur between of illegitimate mitotic recombination or slippage during DNA replication. So I think this is probably mostly caused by transposons and related repeats.
All right. So before we move on to our next speaker, I'm going to ask you a question. So you talked to us about gene editing techniques as it applies to tomatoes. So what are the other plants that CRISPR and other gene editing techniques have been applied to? And also what other traits have been modified using this technique? Like I would imagine disease resistance, pest resistance, or tolerance to environmental changes. Could you share your thoughts on this?
Yeah, so you're right. Quite a few systems have already been modified. So corn, soybeans, wheat, you know, I could go on. There are many that have now been using genome editing to modify particular genes and traits. Disease resistance has been a big one. There's a great study out of China and wheat where I think 4 or 6 homologous genes were targeted to eliminate the powdery mildew pest from infecting those plants, so giving you tolerance to a mildew. There's metabolic pathways that have been affected. There's, for example, in soybean, there's work on modifying oil content and generating soybean that makes high oleic acid content, which is a healthier oil. So I think it's expanding dramatically. I think the challenge that faces us, very briefly, is that not all systems are amenable to genetic transformation. And so we take for granted when we say, well, we have these genome editing tools at our fingertips, CRISPR-Cas9 to make large and small deletions and inversions, genome editing based on base pair changes, base editing, prime editing. All these tools are being migrated into plants, but not all plants are transformable in order for us to actually introduce the editing machinery into the plants to get the edits that we want. And so the biggest bottleneck is actually the transformation bottleneck. And our collaborator at Boyce Thompson Institute, Joyce Van Eck, is a transformation specialist. And this is something that she's been working on for years and has emerged as a key force in being able to address that bottleneck.
All right, we have time for one last question from Rukmini, and then we'll move on to our next speaker. Rukmini, please go ahead, ask your question. All right, she wants to know what would the shelf life of CRISPR tomatoes be?
Yes, it depends on if you're targeting a gene that would extend shelf life, or you're working in a variety that already has extended shelf life. So I'm going to make that question a little bit more relevant to a broader question, which is background dependencies. So varieties have natural mutations. Those natural mutations are called background modifiers. Those modifiers can have additive effects or epistatic interactions with genome edited mutations that you're creating. And so I could make a genome edit in a particular gene that might extend shelf life based on published literature. But if I do that across 100 different varieties, the magnitude of by which I could extend shelf life would vary quantitatively across those 100 varieties because of background modifiers that influence the effects of the mutation that I created. And so it could be that it would affect shelf life in a predictable way, but the magnitude of that shelf life change would vary depending on the natural mutations that are already there and giving you these background dependencies.
All right. Thanks once again, Zach. And I guess we'll continue the discussion on the Slack channel. And at this time, we move on to our next speaker, Dr. Rebecca Leishan from the Branbury Center, also from Cold Spring Harbor.